So now that we've talked about the first half of the citric acid cycle and where carbon dioxide molecules are generated, we're now going to focus on converting our 4-carbon molecule back to oxaloacetate. Now this is going to require you to think about the different high energy molecules we're generating that are carrying electrons and electrons and hydrogen ions. So we've talked about NAD and NADH quite a bit already, and I just want to emphasize again, as I did in chapter 22, that NAD and NADH are going to be involved in reactions that are going to produce carbon-oxygen double bonds. And we saw that in reactions that form carbon dioxide, as well as reactions that took a hydroxyl group, an OH group, and converted it to the carbonyl group, or the C double bond O group. So that gives us an NADH molecule. Now the other high energy molecule that we haven't seen yet is this molecule FAD. Now FAD is going to also carry electrons and hydrogens, but it's going to be involved in reactions that produce carbon-carbon double bonds. So when we have a carbon-carbon single bond, as we'll see in one of the later reactions of the citric acid cycle, these carbon-carbon single bonds can be converted into carbon-carbon double bonds in the same way that an alkane can be oxidized to produce an alkene. Now when we do that, we're going to lose hydrogens because we're forming a double bond, and those 2 hydrogens and electrons are now going to be partnered with FAD to make FADH2. So again, we see where there's an oxidation reaction, it's always partnered with a reduction reaction. So the oxidation of a carbon-carbon single bond to make a carbon-carbon double bond is going to be accompanied by the formation of FADH2. So I'll highlight this again whenever we get to that reaction. So last time we talked about all of these reactions that lead up to the formation of succinyl-CoA. Again, these were going to be where we took our 6-carbon molecule and converted it to a 5-carbon molecule by the removal of carbon dioxide. And then we took immediately that 5-carbon molecule, converted it to a 3-carbon molecule, again because of the loss or the production of a carbon dioxide. So now we're going to take the last part of this reaction and really just talk about the fact that we need to get our 4-carbon molecule back to oxaloacetate. And we're going to talk about the steps that it takes to do that. Now the reason that that's required is because the citric acid cycle, in order to continue, there has to be oxaloacetate present. So if we're going to keep metabolizing glucose by taking the pyruvate, converting it to acetyl-CoA, and adding the acetyl group to oxaloacetate, we have to get back to oxaloacetate. So that's what we're going to talk about in these next slides. So succinyl-CoA is the molecule that we ended with last time. Now this 4-carbon chain is now attached to coenzyme A. And the very first step in this last half of the reaction is going to be the removal of that coenzyme A molecule. Now that's going to give us, now this is a high energy bond. It's called a thioester bond because it's like an ester, only the oxygen is replaced by a sulfur. So a high energy bond that we're going to cleave right here between the C double bond O and the sulfur. Now that high energy bond is going to release the coenzyme A, and it's going to produce for us a GTP. So the energy stored right here is going to be used to add a phosphate onto GDP to make a high energy GTP molecule, which is essentially the same as making an ATP. So don't let the G confuse you. It's the same energy value as an ATP. So that gets us to a molecule called succinate, which you can see is a symmetrical molecule. Now the succinate molecule is going to undergo a process of dehydrogenation. Now hopefully you remember that dehydrogenation is the removal of 2 hydrogen atoms. Now if we do that, we're going to produce a double bond a carbon-carbon double bond. And again, I just told you guys on a couple of slides ago that anytime that we take a carbon-carbon single bond and convert it to a carbon-carbon double bond, we're going to have the involvement of FAD. So the oxidation of succinate to fumarate is going to be accompanied by the formation of FADH2 from FAD. So oxidation occurs here and reduction occurs here. Again, high energy molecule that's going to be used to produce ATP. So 2 hydrogens are removed, they're donated to FAD to produce FADH2. So that's what we see here. Again, we're taking our symmetrical molecule, we're taking off the coenzyme A, giving us our succinate, and then introducing a double bond to produce fumarate. Now in the very next reaction, the molecule is going to undergo hydration. So hopefully you remember this one too, right? So hydration is adding in H2O at the double bond. So that means that one of the carbons is going to get a hydrogen and the other carbon is going to get an oxygen. Now when that happens, we end up with a molecule called malate. So the malate molecule is now going to undergo dehydrogenation itself. Only this time the dehydrogenation isn't between the hydrogens of adjacent carbon atoms, but rather the hydrogen of the hydroxyl group here and the hydrogen on the same carbon. So we saw that in the conversion of alcohols to ketones, right? So this is like a secondary alcohol right here. So we oxidize that to form a C double bond O. Now again, anytime that we've got the formation of, or the conversion of a single carbon-oxygen bond to a double carbon-oxygen bond, we have the involvement of NAD. So NAD is going to take a hydrogen and form and electrons and form NADH, and the other hydrogen will be left as a proton. So we're converting malate by oxidation to oxaloacetate. And again, oxaloacetate is the molecule that we started with at the very beginning. So if you follow that a carbon-oxygen single bond being converted to a carbon-oxygen double bond is going to involve NAD, then you already know that just by this conversion that you're going to be producing a high-energy NADH molecule. So again, we add water into a double bond to make an alcohol. So this is oxidation because we're increasing the carbon-oxygen bonds, right? So there aren't any carbon-oxygen bonds here. We introduce a carbon-oxygen bond here, and then we oxidize again to take this carbon-oxygen single bond to make a carbon-oxygen double bond. And again, that gets us all the way back to oxaloacetate. Now in the next section, we're going to talk about how these NADH and FADH molecules are used to generate ATP. So go ahead and think back about this last half of the cycle before I summarize the citric acid cycle as a whole. So the citric acid cycle is going to utilize an acetyl group that came from our pyruvate molecule. Remember that pyruvate is the product of the breakdown of glucose in glycolysis. So the acetyl group is transferred, the 2-carbon acetyl group is transferred to oxaloacetate, a 4-carbon molecule, to form citrate, a 6-carbon molecule. There are then going to be 2 decarboxylation reactions that remove carboxyl groups to produce 2 molecules of carbon dioxide. Remember that the first molecule of carbon dioxide was generated when pyruvate was converted to acetyl coenzyme A at the very beginning of the citric acid cycle. So these are the last two carbon dioxide molecules generated from any one pyruvate molecule, which is, again, the product of glycolysis. There are then going to be 4 oxidation reactions that provide the reduction to produce NADH, 3 molecules of NADH, and 1 molecule of FADH2. And then there's going to be 1 molecule, or the equivalent of 1 molecule, of ATP generated directly in the citric acid cycle. The rest of the ATP is going to be produced from the NADH and the FADH2 molecules that we saw produced on the previous slides. So the citric acid cycle is going to be driven whenever the cell conditions indicate a deficit of energy. So if there are low levels of ATP and high levels of ADP, this indicates that there is an energy deficit in the cell. So the cell wants to make more ATP and more NADH. Okay? So it's going to be driven whenever these molecules are in low supply. So when energy is needed, it's going to activate the enzymes that convert molecules like isocitrate to alpha-ketoglutarate. And then the same kind of reaction that would convert alpha-ketoglutarate to succinyl-CoA. So low energy states indicated by ADP will drive the process. If we already have high amounts of ATP and NADH, then the cell doesn't need to carry out these pathways because there's already enough energy present in the cell. So the cell has a way of detecting the energy need by monitoring the amounts of these high energy molecules within the cell. So the citric acid cycle occurs in the matrix of the mitochondria, which is the middle part right here, and we'll continue our discussion of the mitochondria in the next section when we talk about the electron transport chain.